Sri Gurubhyo Namaha. Welcome to our online learning initiative, and I welcome you all for the biology class. Last class we saw about the unit reproduction, and now we are going to step into the next unit. Before going into the next unit, I would like to show you some images. Just figure out what it is. So here we'll go. Now you can see an image here. Okay, something like a ball, spherical ball like structure, right? It is nothing but a virus. Okay, it is a virus. And here you can see a double helical structure. It is DNA, deoxyribonucleic acid. So by this time you would have figured it out what type of virus it is. It's none other than the COVID-19 coronavirus. It's worldwide familiar now. So this virus, few decades ago, it was so harmless. You can see it was so harmless due to some factors like induced factor or by the global warming. The virus totally transformed into a harmful virus due to mutation. You can see how it transformed from harmless to harmful. Now stay with me. I'm talking here about a gene. A particular gene was triggered and this has modified into a harmful virus. Number one. Number two, I'll show you an image here. You can see the image, different types of images. Yeah. Number one, a dog and puppies. You can see resemblance. Yeah, something same. It's nothing but the coat color. They have the same coat color. It get inherited from the parent to their offspring. Number two, you can see babies. There are identical twins, non-identical twins, and some variation will also be there, like iris color will not be the same, will be different, hair color, etc. And number three, you can see cows, two cow, one with the same coat color, another one with patch of colors. Yes, so that is a variation. It is not same, there are some variation. And number four, if you see, there are so many flowers, and the flowers, to the surprise, from the same plant, it is of different colors. You can see here yellow color and then mixture of two colors like a blended, okay, blending colors we can see. Number five, you can see a plant, pea plant. It's nothing but Pisum sativum. It was so much familiar in 10th standard. You'd have read in a lesson called Heredity and Variation. Okay. And all these one, why am I showing all these one? Previously I showed about coronavirus, something about gene. And here I'm talking about the coat color, something get inherited from parent to the offspring, and there are some variation among the parent and offspring itself. Okay, so what is that? What is the basic line behind all these things? It is nothing but genetics. So I welcome you all to the world of genetics. Now, in this 12th standard, we are going to see the unit genetics and evolution. This unit is very important. It has a weightage of about 18 marks. In this first lesson, we are going to see about principle of inheritance and variation. So, what is genetics? Genetics is a branch of biology that deals with the study about inheritance and variation. Just see here, it is a branch of biology, okay, which deals with the heredity, the mechanism of hereditary transmission, I mean the characters from one generation to another generation, okay, and the variation of the inherited characters among the similar or related organisms. Okay, when we are talking about the similar or related and related organisms, that is about hybrid also I can say here. So what is hybrid? Hybrid is nothing but the combination of two characters, okay, from the two parents, right? All these things we are going to see in detail later in this class. So we know genetics, it's nothing but the study of heredity. There should be some great person behind the study, right? He's none other than G.J. Mendel, Gregor John Mendel, whom we call it as father of genetics. Okay, it's like 1 mark question, who is called as father of genetics. Then Mendel studied what he studied about the traits. The traits are nothing but characteristics of a pea plant. So there are 7 characters, contrasting characters we are going to learn in this chapter. So what is a trait? A trait is a specific character as I discussed now. So what is it 7 contrasting trait? I'm not going to say all the 7, just 1 or 2. I mentioned here, just check seed color, height, okay, and then the flower color, etc. I'll show you later. So who is Mendel? He was a monk. He was not a great teacher, but he was very much interested in statistics, mathematics, and very much interested to grow the plants also. That is why he took Pisum sativum as his experimental species. Okay, he was very much interested in plants and heredity. So everybody got a word called rejection. Even Mendel's work was ignored by the time. He was also underwent that rejection. So why his work got rejected? There should be some reason, right? The reason is none other than it was too progressive by the time. Okay, when Mendel submitted the work, he was talking about the chromosome, mitosis, meiosis. You can see here. Okay, all these things about molecular level he was talking. So by the time none of them were aware of it. Okay, and one more thing, there is another reason also. They were all so busy in talking about one more scientist whom we call it as father of evolution. Would have guessed by this time? He is none other than Darwin. Darwin's work they were discussing, so they ignored Mendel's work. That was also yet another reason. So a good work will always be recognized. The same way, Mendel's work was also recognized by 3 different scientists. They were doing their work individually and submitted. So it's a very important 1 mark. Hugo de Virus, number 1, first scientist. I said 3 scientists, right? First scientist. And then Carl Correns, Eric von Schermark, 3 scientists. 1 mark question. Name the 3 scientists who rediscovered Mendel's work. Okay, and they rediscovered individually. How many years it took? 16 years. Okay, 16 years after Mendel's death, they rediscovered. Okay, they independently duplicated his experiments and Mendel's work got recognition. So, why was Mendel successful? There should be some reason for this also. So, 2 reasons we are going to take. Number 1, the experimental species, what he selected. It's nothing but Pisum sativum. There are so many plants, why you want to select only Pisum sativum? So, there is also some reason. It is nothing but the size of the plant. Okay, you can see one after the other. Small, easy to grow, and it can produce many offspring in a shorter period of time. Okay, can mature quickly. There are many varieties, as I said, 7 contrasting characters we are going to see. Then it is easy to fertilize. 2 fertilization we can do, both cross fertilization as well as self fertilization. He used, this is another very important reason for his success, quantitative approach. I'll say you an example. I'm going to do a research, okay, and I'm going to have a farm or anything, okay, garden, and I'm going to do only for 20 species or 30 species, okay, and I'm getting a result. I will submit my thesis. Whether my work will get recognized? No, because I'm using only some small number of plants. I'm not using in a huge. Same, I'm going to do the research, okay, for the next set. Some 100 I'm going to take. I may not get the same result, or I may get the same result also. The number, how much you take for experiment matters. So he took more than thousands of Pisum sativum and he did his research and he got good result. In all the research, I mean all the experiment, whatever he took, he got good result and the same result. So his approach, quantitative approach, also plays a major role for his success. I told you about the 7 contrasting character, right? Here I have categorized them depending upon the parts of the plant. Number 1, seeds. You can see your seeds. Okay, I'm talking about parts of the plant. Number one, seeds. Number two, flower. Number three, pod. We call this as a pod, right? And number four is stem. Okay, now under this I've categorized. If you see form, the form otherwise called as shape, how it is looking like. Okay, whether it is a round or it is wrinkled. Then cotyledon color, if you see, there are two different colors. Now just observe this for some 10 seconds. You can see there are some variation among them. Okay, that you call it as contrasting characters. If one is round, another one is not same. It is wrinkled. You can see here, it is wrinkled, right? If one is white color, another one violet red. Okay, simply we call it as violet. That is enough. Another one, if you see, it is flat. Okay, we call it as full. Another one, it is constricted. So there are some variation among them. That you call it as contrasting characters. In next slide, very clearly I've explained here and differentiated one as a dominant, another one as a recessive. So you may have a doubt, what is a dominant and recessive? Here I was talking about contrasting character. Now new thing I'm including, it's not new. The same thing we are categorizing in such a way that a parent, okay, of two different characters, okay, a tall plant, okay, I'll write here, a tall plant when it is intercrossed with a dwarf plant. I've taken the character size. Dwarf plant. What is the character you will get in the first generation? Okay. Based on that only, Mendel said that these are all the dominant character. The character which is seen in the F1 generation, the first generation, are called dominant character. The character which is masked, covered, and not allowed to express is called as a recessive character. So based on that, I've categorized here. So again, I'll recap. 7 contrasting characters are there. In that, we are categorizing as number 1, dominant. Number 2, recessive trait. Right? How are we saying it as a dominant? The character which expressed in the first generation is called dominant. And the character which is not expressed are called recessive character. So one part is clear, dominant and recessive. Another part I'm going to use, okay, I'll write here. This is very important. Phenotype. Phenotype. All these things we call it as terminology. Okay, phenotype. Another one, genotype. Okay. Now, by seeing, I can differentiate. The plant is tall, the plant is short, dwarf, the seed is round, and the seed is wrinkled. Okay, the color of the seed is green, and the color of the seed is yellow. So, by seeing, I can differentiate it. So, what I'm differentiating? The physical appearance I'm differentiating. That physical appearance, we call it as phenotype. You can see here, phenotype is nothing but physical appearance. Now look here and just check. So what are the phenotypes? I'll say one thing, just point out where it is. Yellow color. So here is a yellow color. Yellow is a phenotype. It is a dominant phenotype. What is a recessive phenotype of yellow color? Here it is green. Is a recessive character, right? So these are all phenotypes. I'll write once again here by using different color so you can identify. So it is phenotype. Okay, the same thing I want to express the genotype. In a simple term, what is genotype? Genotype is nothing but their genetic makeup is called genotype. We are saying tall, another one dwarf we are saying. What gene is present in the tall and what gene is present in the dwarf? For that, Mendel took the dominant character, first letter of the dominant character and expressed in a pair. Now it will be very clear for you. P, purple color, right? Purple color is what? Phenotype. What is the genotype for the purple color? The purple color genotype will be capital P, capital P. So as I said, what Mendel did, the genes he is expressing as first letter of the dominant character in uppercase capital letter, and it has to be written in pair. In pair you call it as alleles. Okay, so I'm writing here capital P, capital P. Okay, now how to express the recessive trait? The recessive trait can be expressed like this: small p, one second, I'll erase this. It is expressed as small t, small t. Yeah, listen. Small p, small p. Okay. And here, tall. So, tall. Now, you can say tall is the phenotype. So, what will be the genotype further? The first letter. You can see my cursor here. The first letter of the dominant character should be written in pair. Capital T, capital T. That pair you call it as alleles. The same thing for recessive. How will you write it? Small t, small t. I will write for another one. Remaining all you are going to complete it. Okay. So let me take for yellow color. Yellow color, what again? The first letter I'll take it. Capital Y, capital Y. And here small y, small y. And in exam you'll have a doubt. See, it is green color, so I can use small letter G. No, it is not possible. As Mendel's expression only we have to follow, okay, as how he has given the expression the same way. So if it is a capital letter, the same letter in the lower case you are going to write it. So remaining all these four you are going to complete it. Okay, so now we'll move to the next one. A very interesting thing. Okay, I'm going to show you now. Just observe. Here, unfolding the gene. I'm going to bring out the gene from the cell. Okay, just stay with me. I'll show you. A cell, we all know the simple structure of the cell. Okay, just I'm drawing a rectangular structure of cell. In the cell, there is a nucleus. Okay, in the nucleus, there is a nucleolus, the spherical body. And then there is a thread-like structure, what you call it as chromatid. Okay, and this thread, when it is, that is during cell division, it will be condensed and it will look like this. The appearance will be like this. And this you call it as chromosomes. And this chromosome, something will be there inside the chromosome compressed. I think it is double helical structure. Just guess what it is. A double helical structure in the beginning I showed. It is nothing but the DNA. Okay, it is coiled and it is compressed inside. Okay, I'm just drawing in one arm. It will be present in both the arms. Okay, and this DNA, I'm going to pull it out. The coil structure, it will look like this. The actual structure is a double helical structure when you take it and observe under a microscope. The unwind structure will look like this. Okay, now from here, just observe. Here is the gene. Okay, in this DNA there are gene. So what is gene? Gene is the functional segment of a DNA. So I pulled out the gene from where? From the cell. Okay, from the cell. From the cell, we have seen the chromatids, okay, and then the chromosomes. Chromosome and then the DNA, and from the DNA we have unfolded the gene. Okay, so totally what we saw, I've just introduced the topic, introduced what is genetics, what we are going to see in genetics, that 7 contrasting characters. Okay, slowly terminologies. I've introduced some 4 terminologies. Number 1, what is that? Dominant, recessive, phenotype, genotype. Okay, and I used one another term also, allele. And remaining all we are going to see in next class. Thank you.